Hi ladies, this is a presentation on blood typing. So the question we want to answer is how can blood typing be used to determine parent-offspring relationships? So let's first review. Remember back from our circulatory unit that we were studying blood and we want to know what the components of blood are. So the components of blood are formed elements and plasma. Plasma is mostly water, a little bit of salts, sugars, possibly plasma proteins, and it's also made up of formed elements, which are the red blood cells, white blood cells, and platelets. Red blood cells carry oxygen using hemoglobin. White blood cells are used by the immune system, and platelets can help with clotting. So let's look at the different types of blood that are present. So human blood types are AB, and O. You can also have an Rh factor associated with the blood type. So you could have A positive or A negative, B positive or B negative, AB positive or AB negative, or O positive or O negative. I'll get into more specifics about what that actually means in a second. So blood typing can be used for a variety of different things. Blood transfusion, so receiving blood from somebody else or donating. Paternity cases, criminal investigations, and it's also really important to know for pregnancy. So let's look at the types of blood that are found in the United States. So here's a table showing the breakdown of different blood types in the United States. You can see from this table that O and A are very common. Positive is also more common than negative. So the most common blood type is O positive and A positive. I happen to be A positive. Okay, so let's look at blood types and what these blood types actually mean. So red blood cells express surface antigens, and these surface antigens are caused by a single gene. So in this case, individuals with type A blood, A is a dominant allele. So individuals with at least one dominant allele are going to express A antigens on their red blood cells. Individuals with B are going to also have a dominant allele, so B is also dominant, and these individuals will express B antigens on their red blood cells. When an individual inherits a allele and a B allele, they can express both A and B simultaneously. This is what we refer to as codominant, expressing two independent traits at the same time. Individuals with type O blood actually have no surface antigen, so they have the recessive allele. This is also a really great example of multiple alleles because there are more than two possible alleles. You have A, B, and the O allele. So just a reminder of what codominance is. It means that you're expressing two alleles at the exact same time. So two alleles can be expressed independent of each other. So expressing pink and white at exactly the same time. Note that whenever we do a codominance problem that you want to use the same gene. So in this case, I'm using C's, okay, and I'm using superscripts to denote different alleles. That's going to be really important when we talk about blood type. Okay, here's other examples of expressing two alleles at the same time. Note that there isn't an intermediate here. They're expressing two dominant alleles at the same time. Okay, so let's look at the blood types that are possible and put some genotypes with the phenotypes. So individuals with type A can need at least one dominant allele to express it. So they could have two, so they could be IA, or they could be IA and then have their recessive allele. Okay, that should be a little I. Okay, note that I'm using one letter to denote that because this is one gene. Okay, and I'm using superscripts to differentiate between alleles because there's more than two. Okay, this individual right here would have IB, or they could have IB and then the recessive allele, still expressing the B antigen. This individual, because they are codominant, is going to express both simultaneously, and this individual won't have any surface antigens and will have both recessive alleles. So let's look at how you could draw a Punnett square for this. So if I was crossing an individual with type A blood, okay, type A blood with an individual with type B blood, this is how I would write it if they were heterozygous. So IA and little I. Okay, the other individual would be IB, little I, okay, because they're both heterozygous, so they have at least one dominant allele to express that phenotype, but they also are a carrier for and have that recessive allele. When you put one I allele with one B allele, you're going to produce a child that has type AB blood. If you bring a B allele with a recessive I allele, this individual will express type B blood. This individual would express type A blood. This individual would express type O blood. We also have Rh factors. Rh factors is another gene, and it's another surface antigen that's present on the red blood cells. Individuals that are Rh positive are expressing the positive surface antigen on their cells. Individuals that are negative are not expressing any surface antigen. This is complete dominance, okay, 2 alleles, so you can just use the regular simple dominance that we've been using in the past to demonstrate that on a Punnett square. If I was going to write out the possible genotypes for the phenotypes, I would write big R, okay, they need at least one dominant allele to express the dominant phenotype. Rh negative is recessive. I've also seen that you could do positive, and positive, negative to show this as well, and then this one would be negative, negative. So however you'd like to do it. Okay, let's do a practice problem. So in this practice problem, a man with blood type AB marries a woman with type O blood. What blood type would you expect to see in their children and in what percentage? So pause the video for a second and answer this question. Okay, so an individual with type AB blood, AB, would have this genotype, IA, IB, using the superscripts to denote the different alleles. Individual with type O would have 2 recessive alleles. If I did a Punnett square, so I would get 50% of my children would have type A and 50% would have type B. Okay, in order to have type A, you need to have one dominant A allele. In order to express B, you need to have at least one dominant B allele. Okay, I is recessive, so you need 2 I alleles in order to express type O blood. Okay, so blood typing can be used for paternity cases. So if you can see right here in this table that, if I can get my pen to work, this table right here, here are all the possibilities. So if you cross a mother with type A with a father with type A, you can see possibly what children they would produce from that cross. And you can see that if the combinations don't match up with the children that are produced, you can usually determine that it's possible that the father is not you can determine that it's not the correct father. We're going to be doing a lab looking into this a little bit more next time we meet. So let's look at how we can actually use and analyze and determine what blood type is. In order to analyze and kind of understand what someone's blood type is, either for transfusions or determining paternity, we need to know a little bit of vocab. Number one is antigens. So this is just a refresher from what we've been talking about. Antigens are just proteins that are expressed on cell membranes, okay, and they're very specific for cell types. If you remember back from our immune system unit, we were looking at antibodies. So antibodies are produced and secreted by white blood cells, B cells in particular, and they recognize foreign cells by binding to the antigens on those foreign cells. When an antibody and an antigen come together, a blood clot can result if the antigen is on a red blood cell. Okay, this can cause agglutination or blood clot to be created. So what you want to think about is how can antibodies be used to determine blood types, and why is it important in blood transfusions? So let's look at how this is going to help us determine blood type. So the first thing is with your immune response. So a person will produce antibodies against something that's foreign. Okay, so against any antigen that's foreign. So an individual with type A blood would have B antibodies, okay, because B antigens are foreign to them. So they are going to have antibodies that will tag B as foreign, because it could be anything. It could be some kind of virus or bacteria or something that's not supposed to be in their body. So B is foreign. They're used to A, so they don't want to have any A antibodies. Type B blood has B surface antigens, and in addition, they have A antibodies because A is foreign. Type AB blood, they have type A and B antigens, and they don't have any antibodies. Okay, they don't want to tag something that they normally have in their body. Type O blood, because they don't have any surface antigens, A and B are foreign, so they're going to have A and B antigens, or excuse me, antibodies. It's really easy to mix up the words. Okay. So what you need to do is try to fill out this table. So how you're going to do this, I'm going to do an example for A, and I'm going to let you try to do the rest yourself. So for individuals that have type A, they are going to have A antigens on their red blood cells. I added this column, so you might want to add this column onto your table as well. It'll be helpful to you. Individuals with type A blood will have B antibodies because B is foreign. So what you want to try to figure out is if B is foreign to this individual, who can they donate to and who can they receive from? Okay, so let's start with from. So this individual, because they have B antibodies, could receive from somebody with A blood. Okay. They could receive from somebody with type O blood. Okay, they can't receive from B because there's B antigens that'll bond to their B antibodies, and they can't receive AB blood because AB also has B antigens that'll respond with their B antibodies. So individuals with type A, though, can donate to individuals with type A blood, okay, because they have the exact same type of blood. They can also donate to individuals that have type AB blood, okay, as individuals with type AB blood don't have any antibodies that will react with the A antigens because they're used to having A and they're used to having B. So what I want you to do is pause the video and try to fill in this table. Okay, so I'm going to help you fill in this table. So individuals with type B blood have B antigens. Individuals with AB have A and B antigens. Individuals with type O do not have antigens. For for B blood, have B antigens. A is foreign, so they have A antibodies. No antibodies, nothing's foreign in AB. And type O has A and B. So I'm going to do receive from first. So individuals with type B can receive from B. They can also receive from O. O will not interact with the A antibodies because there's no antigens. Okay, AB can receive from everybody. They are the universal acceptor. And type O can only receive from O. Type B can donate to B. They can also donate to AB, okay, because AB has B antigens on the surface, so it won't interact with their lack of antibodies. Individuals with AB can donate only to AB. An individual with type O can donate to O, AB, A, and B. Okay. You can see that this individual is the universal donor. So what you need to do before next class is test your knowledge at this site and you do not need to do the pedigree.